So now on to neuroglia. So neuroglia, or glial cells, as they're sometimes called, are cells that support neurons. They're smaller than neurons. There may be 5 times as many neuroglia as neurons, or around 100 billion in total. There are 4 types of neuroglia in the central nervous system. There are astrocytes, oligodendrocytes, microglia, and ependymal cells. So astrocytes, also star cells, are the most common glial cell. They have many processes that surround and sequester the blood vessels in the brain, creating a blood-brain barrier. Oxygen and nutrients diffuse from the blood into astrocytes, and then astrocytes share the oxygen and nutrients with neurons while protecting them from any harmful substances in the blood. Astrocytes repair injuries and produce scar tissue. They contain many microfilaments and form a strong mechanical framework to support neurons. They clean up waste products, ions, and neurotransmitters in the interstitial space around neurons. Their functions are many and still under study. Oligodendrocytes, or few branch cells, have processes coming off them that are broad sheets. These processes wrap around neurons and form a protective myelin sheath. We'll discuss myelin quite a bit later. The sheath creates insulation that prevents abnormal spread of electrical impulses, and as we'll learn later, also speeds up the spread of electrical impulses through the axons of neurons. These processes that come off of the oligodendrocytes also tie neurons together. If you look in the picture, you'll see that the processes are on more than one axon of more than one neuron. Areas of the central nervous system that have many axons with myelin sheaths around them appear white. This part of the brain or spinal cord is called white matter due to the white appearance due to the myelin sheaths. Microglia, or tiny glia, are phagocytes. They engulf pathogens if they get into the central nervous system, which isn't common but can happen. They engulf tumor cells, debris, and waste. Only about 5% of neuroglia are microglia, but their numbers will increase if there is an infection in the brain. Ependymal cells line the cavities of the spinal cord and brain. They help produce cerebral spinal fluid that surrounds, protects, and nourishes the spinal cord and brain. Ependymal cells may have surface microvilli that secrete and absorb cerebral spinal fluid. They may have surface cilia that actually, when they beat, move the cerebral spinal fluid through the canals and cavities in the central nervous system. And on this picture, we just see some ependymal cells, and in the scanning electron microscope picture on the right, we see cilia looking very much the way that cilia look when we see a scanning electron microscope picture of pseudo-stratified ciliated columnar epithelium, for example. Okay, so neuroglia in the peripheral nervous system. This is a different crew. There are 2 types of neuroglia in the peripheral nervous system: satellite cells and Schwann cells. Satellite cells surround the cell bodies of neurons in peripheral ganglia, or clusters of cell bodies. They separate the cell bodies from one another. And you see here in the picture, and also in the histology slide, the neurons look gigantic, and the satellite cells, which we see by their little nuclei, are outnumbering the neurons and are much smaller than the neurons. Satellite cells regulate the exchange of oxygen, nutrients, carbon dioxide, and waste product between the neuron cell body and interstitial fluid. Okay, now Schwann cells cover every axon in the peripheral nervous system. They protect the axons and help repair any injuries. Schwann cells insulate axons from each other. This prevents the abnormal spread of electrical impulses, and we will also find out that the insulation speeds the travel of electrical impulses within axons. Schwann cells wrap around some axons repeatedly. This creates thick myelin sheaths. The sheaths protect the axons and increase the speed with which the electrical impulse travels through the axon. And the way that Schwann cells create the sheath is by throwing their cell body, especially the nucleus, over the axon and then over the axon again, and around and around and around goes the Schwann cell nucleus. And as the Schwann cell wraps itself around the axon, the cytoplasm is squeezed out of the layers of the cell, and only layers of the phospholipid membrane of the Schwann cell are left around the axon. So you end up with multiple layers of phospholipid membranes surrounding the axons, and these multiple layers of phospholipid membrane are called, as a structure, myelin. Okay. And this is a big picture of a nerve cut in cross section, and the common pink circles that sort of have a crinkly looking outside, as though we're looking at tiny little like pies, pies like an apple pie, are axons surrounded by myelin. Then the purple nuclei are the nuclei of the Schwann cells. A Schwann cell is only able to surround about 1 millimeter of an axon. Therefore, multiple Schwann cells are needed to myelinate an entire axon. The area of an axon that is found between Schwann cells that is not covered with myelin is called a node of Ranvier. The part of the axon that is wrapped in myelin is called an internode because it's between the nodes of Ranvier. Another way that Schwann cells interact with axons is that instead of one single axon being wrapped by a Schwann cell very tightly, instead, sometimes a single Schwann cell enfolds multiple axons in superficial grooves in its cytoplasm. These axons are called unmyelinated because the whole structure of myelin isn't there, but they are not completely unprotected. They are still embedded and embedded in the Schwann cells and protected by them. So unmyelinated doesn't mean completely just raw and naked and out there. So electrical impulses and action potentials. And this is really a physiology subject, but I feel like an introduction to the nervous system would be incomplete without just a few slides about electrical impulses and action potentials. So neurons, skeletal muscle fibers, cardiac muscle fibers, cells, and some gland cells have cell membranes that can conduct electrical impulses. The impulses are conducted by a change in the membrane potential of the cell. So what is membrane potential? Membrane potential is a difference in the electrical charge on the inside and the outside of a cell due to an unequal number of positive and negative ions on the inside and the outside of the cell. The difference in the number of ions is maintained by the selective permeability of the cell membrane to different ions and by sodium and potassium pumps in the membrane. Okay, in particular, the sodium potassium ATPase pump. The difference between the charge inside and outside of a cell is described as the electrical charge inside the cell. The resting membrane potential in a neuron is negative 70 millivolts inside the cell. When a neuron is stimulated, a change occurs in the permeability of the cell membrane to different ions. Sodium ion channels let in sodium ions if there is a stimulation to the cell. And of course, because sodium ions are positive ions. And the inside of the cell temporarily has a more positive electric charge. Oh, see here, there's a link to action potentials by the Khan Academy. That's really quite nice. If the stimulus is strong enough, the membrane potential may rise to negative 55 millivolts. What has happened is that as there are more positive ions inside the cell is no longer as negative inside. It no longer is negative 70 millivolts inside, it's negative 55 millivolts inside. So it's less negative. At this voltage, voltage-gated channels in the axon hillock will open and many more sodium ions will enter. This is the start of an action potential. The positively charged sodium ions that come in will want to move away from each other and towards any nearby negative charges. Therefore, the sodium ions will spread down the axon in the direction of the axon terminal. The sodium ions tend to disperse, and as the action potential moves down the axon, it tends to peter out and become less strong because the sodium ions are no longer really localized in one spot where they're really clumped up and really, really want to spread. So once they're spread out a bit, there's less of a force moving them to spread out along the axon. But each time that sodium ions reach a node of Ron-Va, they trigger the opening of voltage-gated sodium channels, and more sodium ions enter the axon, therefore refreshing the process. This is called saltatory or jumping conduction of an electrical impulse. So the action potential is created at the axon hillock. The sodium ions pass through the axon underneath myelin sheaths, not interacting with much of anything, just traveling and sort of dispersing. Then they get to a node of Ron-Va. They interact with the voltage-gated channels, and voila, more sodium ions come in, and the action potential is propagated along the axon. How fast the impulse travels down the axon depends on several factors. Larger axons conduct impulses faster. Myelinated axons conduct impulses around 6 times faster on average than unmyelinated axons. The largest myelinated axons conduct impulses at around 450 feet per second. On the other end, small unmyelinated fibers conduct impulses at less than 3 feet per second. It's kind of beyond the scope of this intro anatomy course, but it is interesting to learn about which axons are large myelinated axons and which axons are small unmyelinated axons, to learn what sensory information is traveling through the body fast and which is traveling through the body slow, and which motor impulses are traveling fast and which are traveling slow. It's interesting.